Good evening, everyone. I'm just going to give a minute or so for more people to join. And apologies, I see my name is still Elna Conradi. I will change that. Okay, good evening, ladies and gentlemen. I have the pleasure to introduce Ms. Malishca Perumal this evening. Malishca is a medical scientist here at the Northwest University Center for Human Metabolomics. Malishca is a qualified medical scientist and lecturer at the Center for Human Metabolomics at Northwest University. Her work focuses on diagnostic interpretation and of metabolic defects with a special emphasis on inherited metabolic diseases. At CHM, she is actively involved in the diagnostic interpretation of rare diseases and contributes to ongoing research projects. Before joining the CHM, Malishca completed a medical scientist internship and specialized in clinical biochemistry through the NHLS Department of Chemical Pathology at the University of Cape Town. Her training included work at Groote Schuur Hospital and Red Cross Memorial Hospital, where she conducted extensive analysis on human biological matrices. During this time, she honed her skills in diagnosing inherited metabolic diseases and developed a keen interest in mitochondrial diseases. Holding a Master of Medical Science and Physiology from the University of KwaZulu-Natal, Malishca is dedicated to expanding her knowledge and contributing to the timeliest diagnosis of rare diseases. We're also joined by her supervisor, Sarita Melda. Sarita, welcome to you as well. And Malishca is going to tell us about work that she did during her internship. And the title of her presentation is Comparative Analysis of Urine Organic Acid Profiles in South African Patients with MPV17-Related Mitochondrial Neurohepatopathies. So thank you, Malishca. I look forward to your presentation.
Thanks, Prof. Good evening to all the attendees, and thank you for taking the time to join my CPD talk this evening. So tonight I will be taking you through my internship project conducted at the Red Cross Hospital's NHLS IMD lab. We conducted a retrospective audit to compare the organic acid profiles of homozygous positive MPV17 patients with the aim of identifying a unique urine organic acid profile amongst the patients. This project forms part of a sub-study of the UCT-led MPV17 Natural History Project. So tonight, I'm going to give you a brief overview on mitochondrial diseases with an emphasis on MPV17 in South Africa, and subsequently walk you through our project and findings. So mitochondrial diseases comprise a highly heterogeneous group of disorders in which mitochondrial function is impaired as a result of mutations located either in the mitochondrial DNA itself or in a large number of nuclear genes involved in mitochondrial function. An increasing number of nuclear genetic defects have been identified causing mitochondrial dysfunction, either through the accumulation of mitochondrial DNA deletions or through a reduction in mitochondrial DNA copy number, causing mitochondrial DNA depletion syndromes, the latter of which are autosomal recessive diseases that are characterized by severe tissue-specific DNA copy number. Differences in disease presentations are also found between adult and pediatric patients, where adults may present with one of the characteristic mitochondrial DNA phenotypes, such as mitochondrial encephalomyopathy, lactic acidosis, and stroke-like episodes, also known as MELAS, where pediatric patients tend to present early on with severe complex neurological manifestations, hepatopathy, renal tubulopathy, endocrinopathy, or cardiomyopathy with rapid progression. Since mitochondrial DNA is essential for energy production, a depletion in the mitochondrial DNA content leads to energy deficiency. Consequently, reduced mitochondrial DNA levels result in organ dysfunction due to organs struggling to meet the heightened energy demands, resulting in diverse genetic and clinical manifestations. Clinically, mitochondrial DNA maintenance defects can be categorized as myopathic, encephalomyopathic, hepatocerebral, and neurogastrointestinal. Mitochondrial DNA depletion syndrome 6 is an autosomal recessively inherited disorder that's triggered by pathogenic variant in the MPV17 gene. MPV17 is an example of a hepatocerebral mitochondrial DNA maintenance defect that manifests on mitochondrial DNA depletion in predominantly liver cells. Despite clinical heterogeneity, common clinical manifestations in patients with MPV17 variants include infantile onset neurohepatopathy, cerebral leukoencephalopathy that is seen on brain MRI scans, and metabolic abnormalities such as a prominent lactic acidosis. In rare cases, correlations have been reported between MPV17 and late onset neuromyopathy, with multiple mitochondrial DNA deletions in skeletal muscle, with insignificant or no liver disease. A frequent cause of early childhood death is the progressive liver failure. Many therapeutic approaches have been suggested to treat MPV17. However, the complexity and variability of this disorder makes it difficult to achieve a curative treatment without the risk of relapse. Therefore, targeted approaches to managing MPV17 have been instituted, such as the use of supplementation for decelerating hepatic cirrhosis, dietary modifications, and in severe cases, liver transplants. The MPV17 protein is situated on the inner mitochondrial membrane and acts as a non-selective channel. Whilst its exact role is still uncertain, it has been postulated that it is in some way responsible for regulating transmembrane potential in order to maintain mitochondrial homeostasis, as well as a role in the production of reactive oxygen species. Under normal physiological conditions, the channel opens partially, alleviating excess potential, which limits excessive ROS production. However, if this important modulator is compromised due to mutations, it results in excessive ROS production and the accumulation thereof. ROS have essential functions in cellular signaling, mainly by regulating the expression or activity of many genes and enzymes. However, when at high concentrations, ROS can become toxic to the cell, causing oxidative damage to mitochondrial proteins, lipids, and DNA that may lead to a cascade of apoptosis or necrosis. This leads to mitochondrial DNA instability and depletion, a pathophysiological condition known as mitochondrial DNA depletion syndrome. Mutations in this gene cause severe clinical manifestations that include failure to thrive, hypertonia, hepatosplenomegaly, and jaundice. Patients are either homozygous or compound heterozygous for MPV17 pathogenic variants that are attributed to multiple pathogenic variants distributed throughout the gene. Many variants have been described, including the Navajo variant, and this variant is associated with three distinct phenotypes, namely infantile onset, childhood onset, and classical forms. And until recently, the Navajo variant was thought to be the most prevalent pathogenic variant. Maldo et al. In 2018 reported a common pathogenic variant on chromosome 106, where the nucleotide C had been replaced by T, resulting in an amino acid change from glutamine at position 36 to a termination codon in the MPV17 gene in infants of non-consanguineous Black South African ancestry who presented with neurohepatopathy. Clinical phenotyping, suggestive liver histology, and focused whole exome sequencing led to the identification of this variant in a cohort of 24 South African patients. To date, approximately 73 patients have been diagnosed at the UCT NHLS molecular IMD lab at Groote Hospital with this pathogenic variant. This finding represents the largest cohort of non-consanguineous families with a common variant of MPV17-related disease. This common variant has a predicted carrier frequency of 1 in 68 in the Black South African population in the Western Cape, with an estimated newborn incidence of 1 in 18,622 births. This cohort represents one of the most extensive groups of mitochondrial disease patients with a specific clinical phenotype, all homozygous for a single nuclear DNA variant described in literature to date. So like I mentioned, we are not entirely sure what the MPV17 gene does. However, what we do know is it forms a non-selective channel, but exactly what its purpose is, we can't yet say for sure. There are studies pointing to nucleoside import. Some suggest a role in carbon metabolism, and other studies suggest a role in ROS and membrane potential regulation. So it is located on the inner mitochondrial membrane, and on the MPV17 itself, our pathogenic variant is located at position 36, where the glutamine is changed to not sure if you can see my cursor, but this is where our variant is situated. And this glutamine amino acid is changed to a termination codon, which essentially means that the rest of this protein is not formed. Rest. It also explains why our homozygous patients have such a severe form of this disease clinically. Organic acids are low molecular weight organic substances with one or more carboxylic acid groups and may have keto or hydroxy groups, making them polar compounds. The organic acids are strongly hydrophilic compounds and thus are efficiently excreted by the kidney, leading to their accumulation in urine, also known as an aciduria. Therefore, urine is the biological fluid of choice for the evaluation of organic acid profiles. The interpretation of an organic acid analysis can be a complex task. For a comprehensive evaluation of the obtained profile, and in order not to get lost by the non-diagnostic compounds, it is necessary to keep in mind that multiple factors may influence the observed pattern, such as the clinical status of the patient, diet, plus dietary supplementations, and medication. Moreover, in an emergency situation, sample collection timing may also have an impact on the excreted pattern of organic acids. Organic acid analysis is a key diagnostic tool for the achievement of the differential diagnosis in systemic intoxication, in unexplained metabolic crises, or in the presence of inexplicable routine biochemical findings, such as your metabolic acidosis, your altered iron-iron gap, and hypoglycemia, amongst others. So with all this in mind, I am now going to take you through our project. So the aim of our study was to compare the retrospective urine organic acid findings from South African patients with MPV17-related mitochondrial DNA depletion syndrome 6 caused by a common pathogenic variant. To attain this, our objectives included performing an extraction of urine organic acid reports for all known MPV17 patients with a confirmed genetic diagnosis of homozygosity for the common variant. And where possible, we extracted the raw chromatograms from the above patients to identify slight elevations or patterns of metabolites that may have been missed or deemed unremarkable at the time of reporting. With regards to our experimental data, the original experimental group consisted of 73 MPV17 positive cases confirmed to carry 2 copies of the common pathogenic variant. The age of the original patient group ranged from 11 days old to 13 months of age. And of these patients, only 56 urine organic acid results were extracted from the UCT NHLS mitochondrial disease database and obtained from the NHLS central data warehouse, and through the respective laboratory information systems of the NHLS and the CHM by registered staff members. There was a combination of both males and females of Black South African ancestry. For the pre-analytical phase, it entailed obtaining ethics approval from the relevant Health Research Ethics Committee, GCT. Our inclusion criteria required a positive genetic diagnosis confirming 2 copies of the common pathogenic variant in MPV17, and we collected all our data from the NHLS and CHM. For the analytical phase, I am briefly going to take you through the routine diagnostic procedure followed for urine organic acid analysis at the Red Cross Hospital's IMD lab. So it starts with the extraction of organic acids from acidified urine using organic solvents. These extracts are dried and then evaporated to dryness under nitrogen gas. The organic acids are derivatized, and the TMS derivatives are subsequently identified using gas chromatography mass spectrometry, or GCMS, with ionization by electron impact. Derivatization is necessary to produce compounds that are thermally stable, chemically inert, and volatile, so that optimal separation can be achieved by GC. GCMS is an analytical technique that's used to identify and quantify substances in a sample. It combines 2 powerful gas chromatography and mass spec. Once the sample has been injected into the GC, it is vaporized and carried by an inert gas, like helium, through a long, thin column. And as the sample moves through the column, its components separate based on how they interact with the column's material. Different substances take different times to travel through the column, helping to separate them. Once the components exit the GC column, they enter the mass spec, where they are then bombarded with electrons, breaking them into charged fragments. The mass spec measures the mass-to-charge ratio of these fragments, and this data creates a unique mass spectrum or fingerprint for each substance, allowing for identification. A compound can then be identified based on its retention time and mass spectrum. On completion of a run, the chromatogram is then analyzed on ChemStation, and to aid in identification, we also utilize the AMDIST and NIST libraries respectively. Lastly, our post-analytical analysis comprised of retrieving 50 diagnostic reports from the NHLS laboratory and 6 diagnostic reports from the CHM lab. We were only able to obtain the corresponding chromatograms for the NHLS patient reports, where we subsequently recorded the eluted metabolites and corroborated the reported urine organic acid profiles. These reports were all methodically analyzed and the results discussed descriptively. So the liver, skeletal muscle, and brain are high energy organs and are dependent on mitochondrial oxphos system for energy production, and are therefore highly susceptible to alterations in mitochondrial function. When the mitochondria are compromised, ATP synthesis is decreased, and this can lead to cellular damage. This could explain why these patients present clinically with failure to thrive, jaundice, hypoglycemia, central hypertonia with weakness, and reduced peripheral reflexes, as well as the hallmark hepatomegaly or hepatosplenomegaly. The clinical corroborates what we see in our metabolic investigations, namely the increased liver and mitochondrial markers, as well as a prominent metabolic lactic acidosis. Before I discuss our results, I want to mention that the order of analysis I'm going to discuss now is what we systematically followed in our study, as it was a retrospective audit. However, the correct diagnostic approach to test for MPV17 is to first request a urine organic acid analysis. And if the profile together with the clinical is suggestive of MPV17, then genetic confirmatory testing can be requested and performed. So with that being said, we first pull data from the UCT mitochondrial database. So we're going to look at the GSH molecular IMG process for identifying MPV17. First, the samples underwent PCR, followed by RFLP digestion. So figure 1 shows the separation of fragments on an agarose gel following RFLP, which depicts the C2T mutation in a heterozygous, which is this lane, a homozygous, which is lane 2, and a wild type individual, which is lane 3. A blank control was included in lane 4. Ideally, you would expect to see a wild type or a negative banding pattern in lane 3, with only 2 bands at 262 and 114 base pairs. The 3 bands are for carriers, as one allele has the full 372 base pair amplicon, and the other allele is cut into 262 and 114. And lastly, your homozygous positive patients only have 1 band at 372 base pairs. This blank lane is used for quality control by ensuring there's no contamination from the DNA extraction or reagents that were used during the PCR process. Thereafter, all positive samples undergo Sanger sequencing, which is figures 2 to figures 4. So figure 2 illustrates the sequence electropherogram in a heterozygous individual, and here you can clearly see 2 overlaid peaks, namely your C peak and your red T peak. In figure 3, we have a homozygous individual, and here you will only see the red peak that has replaced your original CT peak. Lastly, in figure 4, you have your wild type individual, and you will see your original blue C peak. So I thought it might be interesting to discuss how we interpret urine organic acid profiles. So this is a profile of a normal individual, and as you can see, there's nothing of significance that immediately stands out. The abundances for the eluted metabolites are all extremely low in comparison to our internal standard. So this peak PDA is our internal standard. For those in the audience who do not know the significance of an internal standard, a quick explanation is that your internal standard is a chemical substance which is added at the same concentration to all your compounds during your sample prep stage and before you run your analysis. The internal standard is typically a major component of the sample or a compound with similar properties, and its concentration is known, and it's used to calculate the concentration of other components within your sample. It also improves the accuracy of your data by compensating for variations that may have occurred during your sample preparation or due to instrument performance. So figure 6 illustrates the urine organic acid profile of a confirmed MPV17 patient aged 1 month old. You can easily spot the variations between your normal profile and this one. And upon introduction to this profile, you immediately note the high lactate and your liver markers. You also note the reverse succinate to fumarate ratio, with fumarate being greater than succinate. You will see your ketones, which are your 2-hydroxybutyrate and your acetoacetic acids. You will see another liver marker as well as your mitochondrial markers. Thiamine was also present in this patient, and we have noted that it is a common metabolite that's excreted when the patients have been exposed to ART therapy. So the total number of urine organic acids that were identified by GCMS consisted of hundreds of components. From information in the Human Metabolomics database, other material that were published on organic acids, and from the knowledge of organic acids acquired over decades in our lab, we established that the most prominent metabolites that were present across all the urine organic acid profiles was the lactate, the liver markers, 4-hydroxyphenyl lactate and 4-hydroxyphenyl pyruvate, as well as our dicarboxylic acids, namely adipic, suberik, and sebacic acids, and their 3-hydroxy components, 3-methyl-glutaconic acid, which is a mitochondrial marker, and the reverse fumarate to succinate ratio. The ketones were also present, as well as phenylactate. So this work displays the systematic review of over 50 urinary organic acid profiles in pediatric patients with MPV17, searching for possible disease biomarkers and providing novel insights into the biochemical features of this disease. An accumulation of lactic acid is generally due to an electron transport chain dysfunction, which decreases ATP production, thereby upregulating glycolysis and pyruvate production, which essentially results in the increased lactate. Importantly, the presence of lactic acidosis is not specific to mitochondrial disorders. Increased venous lactate is also a common profile seen in venous stasis, hypoxia, hepatic dysfunction, renal failure, drug toxicity, and neurodegenerative disorders, as well as many other metabolic disorders. The increased catabolism of tyrosine and phenylalanine occurs exclusively in the liver and can therefore account for the elevated liver markers, namely 4-hydroxyphenylactic acid and 4-hydroxyphenylpyruvate acid that we did observe. This is also in response to the NADH and NAD ratio and may also be an indication of liver damage that is seen in respiratory tract defects. Impairment of the Krebs cycle results in increased ketone body synthesis following meals, and instead of the normal decrease, it primarily due to the channeling of acetyl-CoA towards ketogenesis. During advanced states of fasting, the liver can also convert the end product of mitochondrial fatty acid oxidation, which is acetyl-CoA, into ketone bodies, acetoacetate and 3-hydroxybutyrate, which can then be utilized as an energy source. MPV17 is a maintenance defect, and therefore the mitochondria are essentially unstable and unable to regulate its normal processes. And this in turn affects crucial pathways like mitochondrial beta oxidation, which is why we see elevations in the dicarboxylic acids. 3-methyl-glutaconic acid is an intermediate formed during the metabolic breakdown of branched-chain amino acid leucine. And because of the dysregulated mitochondria, we see elevations in your branched-chain amino acids and therefore the 3-MGC. Under normal physiological conditions, 3-MGC is found in trace amounts in tissues and body fluids. However, increased 3-MGC is capable of exacerbating mitochondrial dysfunction, impairing mitochondrial biogenesis, and causing oxidative stress. The diagnosis of mitochondrial disorders is complex and remains primarily a genetic one. Clinical presentation, biochemistry, histology, family history all provide a role in generating a diagnosis, but genetics is required for confirmation of an MPV17 diagnosis. Diagnosis of mitochondrial diseases in developing countries such as South Africa is especially challenging due to a number of factors. Firstly, the population is highly heterogeneous, and the prevalence of mitochondrial diseases is still largely unknown. Secondly, information regarding the severity of mitochondrial diseases in the Black African population groups is lacking when compared to developed countries. And thirdly, infrastructure diagnostic procedures, such as genetic testing and the interpretation of sequencing data, is in short supply, mainly due to the limited number of diagnostic centers. Importantly, implementation of this metabolite profile analysis at the Red Cross Hospital's IMD lab has facilitated early diagnosis and prompt initiation of empirical treatment within the South African population. Furthermore, we did collaborate closely with the molecular IMD team at Groote Schuur Hospital for confirmatory testing. So in summary, our study underscores the utility of pattern recognition in urinary organic acid analysis through metabolomics, shedding light on the nuanced differences observed in the metabolite profiles of MPV17 patients. This approach obviates the necessity for identifying a single unique organic acid biomarker, presenting a promising non-invasive screening method for MPV17. Understanding the pathophysiology of this disorder and furthering our knowledge of the underlying biochemical and molecular processes involved in MPV17 will contribute to the development of novel therapeutic approaches. So we urge clinicians in South Africa to have a high level of clinical awareness for MPV17, a low threshold for ordering your basic metabolic investigations, such as urine organic acids and serum amino acids, and to liaise with your local chemical pathologists, medical scientists, and geneticists for intermediate to advanced testing to effectively diagnose patients presenting with a potential mitochondrial disease. These are my references. These are some of the interesting resources that I used to compile this presentation. I would like to take a moment to especially acknowledge the people on the screen, my supervisor and co-supervisor, my mentor and the registrars at UCT, the clinicians at Red Cross Hospital, our collaborators at CHM and Stellenbosch, as well as all the referring clinicians, the diagnostic laboratory staff, and patients, parents, and guardians who were involved in this project. And thank you.
Thank you, Malishca. A very nice work, very nice presentation. You are welcome to post questions in the Q&A section. And there's already something here. So Trudy Martin is saying, thank you for a really interesting and informative presentation. I couldn't agree with you more, Trudy. So while people are gathering their thoughts, let me ask you, something that we have discussed, as also a potential marker in mitochondrial diseases is the 4-hydroxyphenyl pyruvate and 4-hydroxyphenyl lactate and the ratio that, use it in a similar way as we normally use pyruvate and lactate ratio. So was that something that you picked up in your patient? Is that ratio perhaps perturbed in comparison to, I don't want to say normal, but potentially other patients with other inherited metabolic diseases?
So when we conducted the study at Red Cross, we were looking at a qualitative side of the organic acids. We did not report quantitative results, so we couldn't calculate the ratio like we had discussed. But it is something that we are looking forward to doing in a new project. So hopefully we will be able to include more of the biochemical aspects and other metabolic investigations to provide a more comprehensive background.
Okay, we have some more questions here. So Tumelo is saying, our first case of MPV17 MN in Limpopo will be presented at the SSIM symposium. Interestingly, organic acid analysis showed liver markers, heavy lactic aciduria, and tiny thymine peak. Have you come across confirmed cases with subtle organic acid findings, and how can the tiny thymine peak be interpreted in the absence of ARV exposure?
So we did see that is a common profile that we were seeing across many of the patients. We noticed the thiamine peaks in 13 out of the 50 organic acid reports from the Red Cross cohort. And it was something that it was a marker that we were using, like an additional mark that we were using for screening for MPV17. These patients also had smaller thiamine peaks, so they weren't obvious like the chromatogram that I had showed of that patient. But there is also a project, Sarita has a PhD student who's also conducting a project on the effects of thiamine on the mitochondria. So I'm not sure if she can comment on its effects.
Yes, Rita, please do.
So yeah, that is actually something I'm very interested in, and I do believe that thiamine tiny bits of thiamine can actually be seen in the urine, and it's not always a marker necessarily of ACT, although most of the time, especially in South Africa, it would be of ACT administration. But it can also be involved in pyrimidine metabolism or in defects. But at those tiny levels, I don't think that is the case either. I do want to look into it, and the fact that the whole one carbon metabolism has been implicated in MPV17 means it might be something that we should be looking at. There's multiple reasons why it could potentially be there, but we really don't know at this stage. I don't think it's anything that we can really comment on at the moment until we've delved a bit deeper into the pathways that is actually affected in MPV17 deficiency. We just don't know exactly why this disease does what it does, because we don't really know what the channel does. So there's still a lot to be investigated. More questions than answers, sorry.
Okay. Another question from an anonymous attendee: how do markers of ferroptosis vary in heterozygous and homozygous affected cells during ordinary metabolism and under induced stress? Do you think this can be used therapeutically? I'm not so familiar with ferroptosis. I think it refers to lipid peroxidation and processes like that. Malishca or Sarita, any comments from your side?
I'm also not familiar with ferroptosis. I'm not sure if Sarita perhaps knows.
Honestly, the first time I've ever heard of ferroptosis was this year during a tutorial. So I'm definitely not the right person to answer this question. But yeah, it's a whole new field for me to still delve into a bit more. I know a colleague of mine has delved a lot into it recently, so but it's how to answer this question would be a difficult one. I don't know, Chris, have you ever heard of ferroptosis before today?
No, I think it has something to do with lipid peroxidation, but if anonymous attendee would like, we can unmute him or her so we can discuss it. But you know, so you've obviously seen some of the routine biochemistry as well, and you know, sometimes we have trouble with patients where we expect or we suspect that they have neonatal hemochromatosis, which is now called something else that I can't remember. So what were the iron profiles like in these patients? Did you see any iron profiles? Do they have signs of iron overload or not? Anonymous attendee, we'd be very happy to hear your thoughts on this. Okay. Any other questions? I don't see anything. Malishca, sometimes we get this profiles with, I don't want to say, well, you get several aberrations and then also tiglioglycine. And I've often wondered about these, and I've done some reading on it. And apparently tiglioglycine is also a marker of mitochondrial stress. So is this something that you saw in your profiles or not?
No, tiglioglycine wasn't a marker that we picked up across any of the profiles.
Okay.
It was on a profile that we saw a few weeks ago of I think the baby was a couple days old, and the baby was at Donald Gordon's hospital in Joburg, and it was a much more exacerbated profile than we've actually seen at Red Cross Hospital, sorry. So there were a lot of markers, and I'm not sure if the disease progression has something to do with what we see, or if it's just the catabolic state of the patient at the time of sample collection.
Okay. And you mentioned something about liver transplant. So is this an option in these patients?
It was something that we spoke to the referring clinician about for that specific patient that we saw a few weeks ago. And the doctor did say that because the patients present with liver failure as well as an encephalopathy, liver transplantation is not a a treatment in South Africa currently. They are looking for options, but because the baby was such a severe form of the disease, they didn't want to or opt for a liver transplantation.
And in literature, patients that underwent a liver transplant, what was the outcome like?
It was a curative. I think it was more overseas or with other variants, not our variant, our South African variant, but they did have liver transplantations, and it wasn't curative, but they were progressively a lot better than what they previously were.
There are some reports, yes, Malishca's right. There are in milder forms and milder mutations. Our mutation is unfortunately a complete knockout mutation. It means that instead of having a channel that slightly opens or slightly closes or struggles to open or close or is slightly defective, our channel doesn't exist. So our presentation is extremely severe in all of our cases, and having a liver transplant might, it's very difficult to know whether it will work, how well it will work, and what the downstream effects will be, because you might save the liver with this, but the neuropathology is still going to persist. So just replacing the liver in a multi-system disorder might not be enough. It's very difficult to say.
Yeah. Okay, we've got another question here. This is a nice question. So how long does it actually take to interpret these types of profiles? Is it a quick interpretations? Can it be automated? Malishca, I think you should give it a try.
I can. So we do have a process that we follow, and that if the patient or the clinician, so if the clinician calls in and explains the clinical to the lab, they can request a stat sample. So the sample, as soon as it's received at the lab, we put it on the first available run. And a run generally takes per sample, well, at Red Cross, it used to take about 40 minutes per sample to be injected and to receive a chromatogram. So if it is an emergency situation, we have actually released results within 24 hours. And once we have a metabolic result, we can then call the clinician and tell them that this is what we suspect, and this is what we are seeing on the profile, and this has been seen previously in this disorder. And thereafter, we advise the clinician to seek genetic confirmatory testing. And it is the same procedure is also followed there. So then the clinician generally contacts Sarita, who is the senior medical scientist at the IMD lab, and she also includes that sample into the first available batch.
Sarita, you wanted to add something to that?
No, I mean, that absolutely answered it. I actually, on a different point, wants to just say something else. Malishca gave a really, really good talk. I just want to add that MPV17, our clinicians kind of, once you've diagnosed one case, they always say that they can, after 2 or 3 cases of these patients, one of our GIT specialists once told me she can literally spot an MPV baby when they come through the door. So they've got a very typical clinical presentation, and although urine organic acids is a good starting point if you don't know what you're looking for, once you start recognizing these patients, it is very, very quick to sometimes spot them. So we do offer genetics as well without the organic acids. If there's any clinical suspicion of MPV17, I don't mind running the genetics. So we don't necessarily have to wait for organic acids, which can sometimes take a while. And if you have to do the organic acids and wait a week for results, for instance, depending on how busy the lab is at the time, and you still have to wait 2 to 3 weeks for a genetic result, it becomes very long. So I don't mind in cases where there's a fairly good clinical suspicion of MPV17 to get genetics requested.
Okay, yeah, let me just also comment on that particular question. So just in a bit more of a direct way, if it's not a stat sample, then you typically take more than 24 hours to run the sample. Then the data needs to be there's a secondary step that, you know, where the peaks need to be annotated and so on. That would probably take another day in the batch. And then when it comes to the interpretation, I think a person that is experienced in metabolic diseases, that has to interpret complex patients. So this is where we typically look at organic acids combined with amino acids and acetylcarnitines, maybe other tests, and where there is extensive clinical information supplied. I think in such instances, an experienced metabolic specialist can interpret about 20 to 25 profiles a day. So it is fairly slow. If it's less complex, and if there's no clinical information, then it can take a little bit shorter, but it's not a quick thing to do. The other thing is if we have instrument downtime, then that can slow down the process quite significantly. Or if something needs to be repeated, it means that you need to start from scratch, and it's again 2 to 3 days to do the analysis. So unfortunately, it does take time. I think yes, we are, particularly on the analytical side, we're working a lot towards automation, and I think our analysis has now really improved because of automation. And I do think that it's possible in the interpretation side also. In the future, we're probably looking at AI interpretation. That's already happening in genetics at the moment. But I'm nearing retirement, so, well, okay, not quite yet, but I'm hoping that, you know, I'll be able to finish before the machines take over. But for some of you, I'm not so sure. So yeah, so just make sure you've got good other skills that you can apply. But that's it in a nutshell. Let me just quickly see. There's just some other discussions here. Yes, Sarita.
Just to also mention that, like you say, it can take about an experienced person about 20 to 25 a day to analyze, but it also sometimes means that whoever is analyzing it sometimes or especially if it's not an experienced person looking at it, needs to still check it with a second person and discuss the case and read up. In complex cases, read up on things. So it really does, it can take even longer than that. And especially in the site sector where we see a lot of referrals all the time, that you do have cases where you can do 1 or 2 as stat samples, but you also can't treat every single case as a stat sample yes. Because it's just not possible with the volumes that we have in our laboratories. So in cases where it really is stat samples, like Malishca mentioned, it can be done, but it's not feasible to treat every single sample like that because we have limited staff, especially in the state sector. We've got limited staff doing lots of samples and needing to interpret lots of samples. So when it's taking away from other cases that may well be stat samples, it also becomes an issue. So although we try and do it as fast as we can, as many as we can, it does need to be remembered that that's unfortunately the scenario.
Thank you. I've also just an open invitation to anyone who wants to come to the lab and see how this actually gets done. You're more than welcome to come and visit us. Many people have visited us in the past, and we'd be very happy to show you if this is something that you're interested and you want to set it up in your country. If you're from outside or in your province or so, you're very welcome to come and visit us, and we'll be happy to assist you. Okay, I think we have pretty much answered everything. So Malishca, thanks again for a lovely presentation.
Chris.
Oh,
sorry. There's a hand up in the audience. Oluwaku Nto Izima, I'm going to allow you to talk so that you can ask your question directly.
Okay.
Okay, good evening all. I actually don't have a question. I really commend the presenter. I thank her for the presentation. It was elaborate and clear, even though most of the slides had so much information, but she was able to summarize and give us good detailed information about what she had. So thank you for the presentation.
Thank you very much for that, and I hope clinicians who have attended is now well equipped to recognize those MPV17 patients, and please send those samples to us so we can make the diagnosis. Okay, I think we have answered everything. Again, Malishca, thank you very much. Sarita, also thank you for making yourself available for the panel. Thank you everyone for attending. And before I forget, also a big thank you to Pfizer, who sponsors this series. The sponsorship is really a donation. They do not influence the content of the program in any way, and they do not influence the selection of speakers. And we thank them for their contribution, and thank you to you for attending, and we look forward to seeing you next week. Goodbye.
Thank you. Bye.